Hello, everybody, and welcome to the Fungal Files Podcast. My name is Leo. I'm joined by Blessing and by Bryce, and today we're going to talk about Coccidioides Immitis, the pathogenic fungi which causes valley fever. Before we do that, though, we're going to talk a little bit about, what is the perception of fungi and fungus? How do people feel? When I think of fungi or fungus, I think of shows like The Last of Us, which sort of frames fungi as this terrifying, world-ending pathogen. And then on a much lighter note, I think of things like Super Mario Bros., where they use mushrooms as these friendly power-ups and they have iconic characters. And then I don't know if you guys remember a few years ago on Netflix, there was the documentary Fantastic Fungi, which talks about how fungus have this, you know, intelligent network and all these ecological benefits. So kind of interesting. So what is fungus? Fungus, they're eukaryotic organisms that include mold, yeast, and mushrooms. Approximately 1 billion years old, and there's 2.2 to 3.8 million species. Immitis is interesting because it can take on a dimorphic form, which includes both. I'm going to hand it over to Bryce, who's going to talk about the history of valley fever.
So speaking of Mario, that Waluigi-looking guy is Alejandro Posadas. He first discovered coccidioides, which is the description of the disease, in 1982. Dr. Rixford and Dr. Gilchrist discovered the pathogen itself. Okay, so early tracking of valley fever was in the 1930s. This was just monitoring the outbreaks, clusters of cases. This disease has expanded beyond California to Nevada, Arizona, Utah, New Mexico, and even parts of Central and South America. Valley fever is found in hot and dry soils. And what happens, how we get sick is when the soil is dispersed. People breathe in this fungus, and then it develops into spherules, and it's what makes us sick. And then when we get sick, we have symptoms such as fever, cough, chest pain, fatigue, shortness of breath, sweats, and a rash. These symptoms for 60% of people are very mild or not at all. For 30%, you get a pneumonia. Again, pneumonia is serious. It can lead to hospitalizations. And then for 10%, it is very serious to things like it affects the central nervous system. Now, the groups at highest risk are Black pregnant women, immunocompromised people. Now, when people are sick with this, the way that we diagnose this disease is with PCRs. Just gives us the DNA sequences, serology, cultures. That's the best way to find Howie is through cultures. Another way is microscopy, where they look at the sample and they look at the spherules in the specimen. Another way is by chest imaging. There are challenges to this as well. Sometimes it is misdiagnosed as a bacterial pneumonia or even cancer. This isn't a problem because it can lead to delayed diagnosis, which leads to more serious. Thank you. And then overview of the issue. So this is important because of it is starting to spread more northward to the more northward states, and this is due to a warming climate, in part, and then also due to increasing population. As you know, the soil is dry, more opportunities for the fungus to spread. This is important because more people are getting more sick, more people are dying, and it's going to be more costly. Per case in 2010, it was upwards of $400,000 for serious cases, and in 2090, it's going to be $620,000. In 2010, overall, as a nation, we spent $5 billion in treating this disease. In 2090, we will be spending upwards of $15 billion. I am now handing this over to Leo.
Awesome. Thanks so much. Really, three things that we should look out for when dealing with pathogenic fungi. That's going to be virulence, antifungal resistance, as well as pathogenicity. And so virulence really just refers to the degree of damage that this can cause. How quickly does it spread? how strongly it triggers inflammation, and how severe the symptoms can be. Antifungal resistance refers to the mechanism that reduces the effectiveness of antifungal drugs. And so, resistant strains are going to survive medications that would normally stop them. And then pathogenicity is the ability for, the fungus to establish disease in the first place. Here are the genes and pathways that are involved in all three of those factors. The first one is CPS1, followed by SALGP, ERG11 pathway, and MDR. These all play a role in whether or not the infection is established, can survive in the host, is responsible for antifungal resistance, and or treatment failure. All this really understates and highlights the importance of genomic analysis in order to improve diagnostic surveillance, detect resistance earlier, and compare strains across regions or outbreaks. Now I'm going to talk a little bit about our data set, right? So we extracted data from NCBI's BioProject. All of this data was processed at AROP Laboratories in collaboration with the University of Utah, and our FASTQ files were accessed through the associated SRA biosample records. The study that we referenced had 49 total clinical isolates, all of which were existing in the Southwest. For this project, we analyzed 23 of the 49 that were identified as Coccidioides Immitis. All samples were sequenced using Illumina pair-end platforms. Now, the reference genome that we chose was just the generic reference genome for Coccidioides Immitis, the reference strain, and we downloaded this and uploaded it into Galaxy. So we wanted to be able to take this raw clinical data and get to the point where we could do variant calling. Additionally, and this was just sort of added gravy or added bonus, was to be able to look at some of these pathways and be able to tell if we could identify regions that would contribute to antifungal resistance, pathogenicity, as well as virulence. And so these were the bioinformatic techniques that we employed within the Galaxy platform. It was really a 7-step process, which included FastP, FastQC, MultiQC, Minimap, SAM tools, BCFT tools, all of which are available via the Galaxy Toolshed. Here is an overview of our Galaxy workflow process and our pipeline that we employed. And here are some of our results, right? So our quality control results showed strong sequencing performance across the 23 samples. Additionally, we saw that there was only minimal overrepresentation of sequences, indicating that there was no major contamination or adapter issues. Overall, the data quality was robust and suitable for accurate downstream alignment, as well as variant calling. These next images that I'm going to go in and show you are through BCFT tools, where we were able to do variant substitution plot, and we were able to look at the balanced distribution of the SNP types across the data set. One of the added bonuses, or one of the other sets of analysis that we were going to try and run, was to look into pathogenicity, antifungal resistance, as well as virulence. And so I was able to go ahead and access through NCBI FASTA files that were related to those pathways and genes that we referenced earlier. And then I was able to use BLAST across our 23 clinical isolates in order to see if there was a match. In this case, it was unsuccessful, but it was still really neat to be able to try it. And this is an analysis that we can possibly try and use later on. And now I'm going to hand it over to Blessing, who's going to talk a little bit more about Coccidioides, the GitHub pipeline that we used.
Yeah, thank you. So the motivation behind the Coccidioides was to find an alternative approach that can be used to improve variant detection. And this can be done by, assigning species for the Coccidioides. And in cases where the standard pipelines, struggle. And how this approach differs to standard variant calling approach is that it starts by adding, it starts with an automated species detection approach, which, decides between C. immitis or C. Pussadis. And this process is done before the variant calling step. And it also uses species specific referencing, masking, tailored processing, as a way to reduce false positives from the results. And also, the end product is a combination of list of VCFs and consensus faster. And the next part of this was, we cloned the GitHub repo. We tried to reproduce the workflow locally, and that involved the resource prep workflow and the variant calling workflow that generates the VCFs and the consensus faster files. And in the GitHub repository, the authors have, they outline instructions to follow to be able to, um, do the exact same thing they did, which could be done either locally or can be submitted as a slum job, whichever works for anyone willing to reproduce. And the dependencies majorly on Nextflow, a container platform like Docker or Singularity, and in this case, we used Nextflow and Docker, and downloaded other necessary dependencies, for the operating system in use. And the end product of that was we had the consensus files, the consensus faster files, that just like they did because we used their own data, and also built on data the Kraken database that were built locally on my computer. And the next phase of it was, each of us experienced certain challenges in our individual stages, and a summary of all the challenges. We are new to the field, and there was lack of experience in being able to get result as quickly as an expert would have, and also there's no single standard method for analyzing cookie diodes. So we were trying a bunch of things, before we could arrive at, the best approach for us. And the last thing was, there were also very few publications available to provide a clear and general path to follow in being able to, conduct this research properly. So I would hand over to Leo.
Awesome. Yeah, we're just going to go finish up on just future directions and plans. So I think the big thing was just time. We didn't have a lot of time to run all of this. We're also not bioinformatics specialists. So for us, it was a lot of trial and error learning as we go.
Well, I would say for the future, particularly in reproducing what was done on the Coccidioides data, have it in mind to prepare to have, a very solid computational power, because that would help with, processing the files faster, because it takes several days to process these files, and also prepare to work dynamically, um, because some of the instructions in the GitHub repo, they were valid at the time of publication, but if you try to reproduce, a couple months later, things change, and you have to be ready to, find your way around those things.